Thank you for giving me the honor of introducing Dr. Harminder. He needs no introduction, but he is currently the chair and professor of ophthalmology at the Queen's Medical Center at the University of Nottingham since 1994 and joined head of the service since January 2018. He's also honorary professor at Universidad de la Civil in Lugano. And he was also made by Queen Elizabeth II, Commander of the Most Excellent Order of the British Empire, a very, very high honor. He's been editor-in-chief of many journals, including the Eurocornea Journal, and he was editor-in-chief for the British Journal of Ophthalmology from 2007 to 2014. He has more than 300 peer-reviewed publications, and on a personal note, when I was doing my fellowship, I had a strong interest in setting up a chemical burn project in our ICU, and I used many of his publications, including his classification, really help us with the project. We also use many of his manuscripts to understand high-risk graft and the role of using T-cell inhibitors. So I think of those 300 publications, many of them are really highly clinically useful for people that are in practice of cornea or just starting practice of the cornea. So I recommend everyone to read the publications. He has other landmark publications, such as the understanding of the limbal corneal area and the role of wound healing. So with that, it is a real honor, and thank you for your time. We know it's late in the UK, and we appreciate having you here in America.
Thank you. Thank you very much. And I hope that our hacking problems are behind us. It's a pleasure and honor for me to be here and to take you through the story of the pre-Descemet's layer or the Duer's layer and how it has become so important in our understanding of not only corneal surgery, but corneal pathology. So, let me see if my slide's going to move. So these are my declarations of interest, and we'll start with this simple DALK procedure with a type 1 bubble. You see the air bubble is separating the what was believed to be endothelium from the rest of the stroma, and you cut the bubble anterior wall. You put in a spatula. Nowadays we fill it with viscoelastic, and then you cut into 4 quadrants. Then you cut the quadrants out, and it was said we are exposing the anterior surface of Descemet's membrane, to which you attach another cornea from which you have taken away the Descemet's membrane. So this is how it started. We were all doing this procedure. So I'm going to give you just a few slides on how the discovery was made. The first thing we realized that if you look at the Descemet's membrane surface that is exposed over here, it was quite strong. You know, you could bounce an instrument or a forceps off the surface. It didn't appear to be as delicate as a Descemet's membrane. Then in the lower picture, you can see that when you're doing a penetrating graft, when you put your needle, we are advised not to come out through the full thickness of the cornea, but to come anterior to Descemet's membrane. If you can see my arrow, just behind the needle is an edge, which was told to us is the Descemet's membrane. So just bear that in mind as we go to the next one. Sorry for this noise in the background, but here you can see that from a donor cornea, the Descemet's membrane is being peeled off, and see how easy it is to peel off the Descemet's membrane from the back of the cornea in preparing the tissue for DALK. Now just keep that in mind as we move to this next slide. Now here, see, I've taken the Descemet's membrane off, but I can still see an edge. And the question was, how come there is an edge, and what is actually causing that edge if it is not the Descemet's membrane, because we have removed it? So there was a clue saying that there is something else over there. And sometimes when the bubble did not go as far as the...
Sorry, I can see.
Sorry?
Sorry, I can see.
Yeah. If the bubble does not go as far as the refined mark,
Sorry, sir, I can see.
then
I
we found
want
there are
fuck
strands
with your sister.
extending between the
I want fuck with your sister.
between the posterior cornea and the anterior surface of Descemet's membrane. Now, when we remove the Descemet's membrane, it is very smooth, but in this case,
Your sister are here with
there are some strands between the posterior surface. So if you remove Descemet's from the deep surface, there are no strands. But if you remove deep surface from the Descemet's, you see the strands. So clearly, we're not working in the same plane. So on the basis of that, we hypothesized that there exists a distinct layer in the posterior stroma.
Ugly meeting Coolera better not get into anything. Go to the fucking motherfuckers.
So we did some experiments to simulate DALC in the human eye bank eyes, and we published these results very early on in 2007. Now, let's show you those experiments. So the first thing is when you inject air in the human cornea, what you will see is that you can create a bubble which starts in the center away towards the periphery. And this bubble does not extend as far as the periphery. It is a centrally located bubble, and we call this the type 1 bubble. But in some occasions, what happened is that we got a bubble that would start at the periphery, and you see how it starts at the periphery, and it comes towards the center. And then it occupies the whole of the posterior surface. And also notice how the air moves in a circular manner along the periphery. See, another bubble is starting at the periphery, and then these two bubbles will meet to form a large bubble, which is very different from the first one. So we call this the type 2 bubble. And occasionally you got this, which is the type 3 bubble, where you can see there's a type 1 or the mixed bubble. There's a bubble in the center, and then over here there's a small crescent of the type 2. Here there's a large central bubble. You can see a large one over here, and then a horseshoe-shaped type 2 bubble. So these were what we call mixed bubbles, where we're seeing two kinds of bubbles happening simultaneously. So in the past, in clinical practice, we always saw these double bubbles, and we thought it was due to the split between the banded and non-banded zones of the Descemet's membrane. That was the explanation provided. But we were able to show that it is something different. And this is the slide really where we were able to do that. So here is a type 1 bubble from which you can actually peel off the entire Descemet's membrane, and the bubble is still intact. So it tells us that the anterior wall of the bubble is impervious, this is to air, and you can take away the Descemet's membrane completely without the bubble deflating. And if you do histology, what you find is there is never a split. Whether you take the Descemet's membrane from a type 1 bubble or a type 2 bubble or from a mixed bubble, there is never a split in the banded and non-banded zone. See, here's the banded zone and the non-banded zone of the Descemet's membrane. They're always intact. So clearly there was air that was accumulating behind the Descemet's membrane, but also behind another layer in the posterior cornea to create the type 1 bubble. Now here's an example of a complete type 1 and type 2 bubble, and you can see inside there is the type 1 bubble with the pre-Descemet's layer, and the outside you can see the type 2 bubble. So this is showing you how you can get complete both type 1 and type 2. So it defined what the air was doing in the human eye when we were injecting to create bubbles for the DALC operation. Now, why is this clinically relevant, or what is the clinical relevance of this? So imagine if you get this situation on the top over here in a human eye, and you are going to do a paracentesis, you will burst the Descemet's membrane straight away. So if you can see this happening, you will not do a paracentesis until you have deflated the air from the top. But in the bottom picture over here, if you know that there is a type 2 component on one side but not on the other, then you will not do a paracentesis on this side. but you will do a paracentesis on this side and you will save the descemetes from bursting. So that's the clinical relevance of this surgical anatomy. But there's something else also very interesting. So now I hope you can hear me. So what you can see is that over here, when you inject air, it follows a distinct pattern in the cornea. You get radial spokes of air moving to the periphery in some cases. In some cases, you get this cracks in the glass like pattern. And then sometimes it's very diffuse and sometimes a combination of the two, some of those cracks in the glass pattern and a diffuse spread of air. So the earliest movement of air is quite different. And this picture over here looks very much like the ICK pattern that we see when you have microbial infection of the cornea in the absence of the host response. And this we have shown in the past represents the migration of bacteria or other organisms along the lamellae of the cornea. So if the corneal lamella is very compact, air will make this movement. But this is the more important part, where once the air reaches the limbus, it moves in a clockwise and counterclockwise direction, as you see, circularly along the periphery till it completes the circle. And then the air comes in. And when you do histology, you can see that the separation of the lamella are much more along that circumference and much less in the center. So this peripheral circulation of air that you see is a very unique feature in the cornea and is telling us that the microarchitecture of the cornea in that region is different from the rest of the cornea. And if you look at the mechanisms now, you will see that air, once it accumulates at the periphery, it then comes to the center. Tiny bubbles form over there. Those bubbles will then accumulate to form a small central bubble and forms the full-blown type 1 bubble. Sometimes it forms small type 2 bubble, always at the periphery. It always forms at the periphery, and then it goes across to form a big one across the whole of the eye. What you also see is that in the videos, you may have noticed there's a lot of air that escapes as tiny bubbles from the periphery, and that's quite relevant, and we'll come to that shortly. Now, how is this clinically relevant? So we know that the cornea is prolate, the center is convex, and the periphery is flat. Now, this flat area, as you see here also on topography, here and over here, you will see in, this here, you see that in the shape of the cornea. This flat area, this flattening at the periphery corresponds to that 1.5 millimeters of the peripheral cornea where the air moves in a circular manner and where the microarchitecture is different. So it is telling us that the thicker peripheral cornea has a different lamellar arrangement than the central one, and that's why it is able to hold the cornea, the central curvature and the peripheral flattening. And we also know that the type 1 bubble will extend from the center to the part where it starts to flatten, and it doesn't go beyond that, although the layer does extend well beyond that. Now, this was another thing we found in the surgical anatomy, and let us see if this video will work. If it doesn't, what I'm going to go is I'm going to come out of the slideshow, and I'm going to play this video over here, and hopefully it should work. So, okay, it's not going to work, but what it shows is that, so on this computer, the QuickTime videos are not working, is that the bubble will burst at a pressure of about 700 millimeters of mercury. So that is quite a high pressure. It's able to withstand that much pressure, the pre-descemetes layer. And on the basis of that, again, this was a video which unfortunately is not going to work over here, but let me try, was that this is Dr. Ahmed Zaki, who he creates a DALC operation, and under the pre-descemetes layer does the complete DALC operation, phacoemulsification with implant, and in one case even a vitrectomy. So showing that because the pressure the ability to withstand high pressure is so much with the pre-Descemet's layer, you can actually complete a DALC operation underneath that. I really apologize because I had to switch computers to try and get rid of the hackers. And on this computer, the software is not there to play some of the videos. But this is what you can see, the outcome of some of these DALC triple procedures. And when we do scanning electron microscopy, now back to surgical anatomy, you can see this is the endothelium. And this over here is the pre-Descemet's layer. And this is a side view, that is the endothelium with the endothelial cells and the Descemet's membrane. And here you can see the pre-Descemet's layer, the edge of the pre-Descemet's layer along, and then beyond that are the lamellae of the corneal stroma. On transmission electron microscopy, again, just to show how the Duer's layer, the pre-Descemet's layer, and you can see those strands extending from deep stroma. Here is the pre-Descemet's layer, very compact, and that over there is the Descemet's membrane. And on histology, all the features, these have been well described on electron microscopy. Now, clinically, it clearly shows that there is this plane of cleavage between the deep stroma and the anterior surface of the pre-Descemet's layer. And this plane of cleavage is very, very important to understand, because what you will find is if the air bubble does not go as far as your trifine mark, as you can see in this video sent by Mr. Hardik Parekh, you can actually lamellar dissect with a spatula and go all the way to the periphery to your trifine mark, because that cleavage plane is easy to exploit. And you will see these strands which you can then cut or trim as you are exposing the anterior surface of the pre-Descemet's layer to the periphery. So knowledge of this pre-Descemet's plane of cleavage allows you to do all of this, and you know the layer itself is so strong that it's not going to rupture on you during the operation. Not only can you actually separate it with a spatula, but again, this video is not going to work, I think, here, because this is Rishi Swaroop has shown. They tried a DALC with viscoelastic, but that viscoelastic didn't work. So they physically, having cut really deep, were able to peel off the whole of the anterior stroma from the pre-Descemet's layer, just physically peel it off, exploiting that cleavage plane. You cannot do that with a Descemet's membrane because that much pressure will cause the Descemet's membrane to rupture. But bacteria and fungi also exploit that cleavage plane. And here you can see this paper by Liu et al. To show that fungi will migrate along the pre-Descemet's cleavage plane when you get keratitis profunda. So in pathology also that cleavage plane is being exploited. But this is something which I will tell you is a landmark discovery, and it will change our thinking completely. It was first published by myself and my colleague Dr. Said as a case report, where you found that a case diagnosed as Descemet's membrane detachment, as you see over here, was treated with air and it didn't work. But if you look very carefully, there are two lines. And then further in the sections, you see that there is this line here, and then you see another line here with a torn, rolled Descemet's membrane. So the pre-Descemet's layer was actually separating spontaneously without injection of air in Descemet's membrane detachment. So this index case allowed us to do a very big study in which we have colleagues, as you can see, Rajesh Sinha from India, Susan Jacob from Narayan Netralaya, Rajesh is from All India Institute, Mohammed Kenavi from Egypt, and Frick is from South Africa, Ian Scott and Dahlia Syed from Nottingham. So together, we published this paper in AJO. It's been accepted only last week, so it will be a few days before it's actually published. So what we are all used to seeing is Descemet's membrane detachment like this, where the Descemet's membrane detaches, right? Very curved and wavy line. But what we will see, and this is a summary of the whole paper, is in this slide, that you get three types of Descemet's membrane detachment, just like the three types of bubbles. Type 1, where it is the pre-Descemet's layer and the Descemet's membrane, and so like a straight line, like the chord of a circle, very taut straight line. Or it can be type 2, which is this double contour line. You always see on OCT two lines of the desmoid membrane. One is from the back of the membrane, and the other is from the endothelial surface with the difference of aqueous between them. And this is a type 2 bubble and is also a type 2 detachment. And you will occasionally see this, which is a mixed one. You get the pre-desmoid layer over there, as you see, and then this curvy desmoid membrane. So we have missed this all along, that desmoid membrane detachment often includes detachment of the pre-desmoid layer. And we have 41 cases to illustrate this. 5 of them we have shown even by histology. So clinically, if you see a very straight line like you see in the picture in the middle, that is a type 1 or a pre-desmoid layer with desmoid membrane detachment. This is how it is, straight line on OCT. And this patient had a cornea transplant, so you can see that the pre-desmoid layer is on the anterior surface of the desmoid membrane on the histology. And again, there are no cells in it, but there are cells on it. And this is the endothelial surface over here. If you see this other patient over here following a graft, exactly the same thing. And remember the strands I was showing you from the deep stroma extending to the anterior surface of the pre-desmoid layer. You can even sometimes see those strands on OCT. Again, you can see the desmoid detachment is a straight line. And on histology, we could see mostly that straight line, there's the pre-desmoid layer. But here, you can see there are some cells infiltrating. There are no mononuclear cells anywhere, but the keratocytes will migrate into the layer and cause scarring and contracture of the layer the longer it is. So that's a type 1 kind of desmoid membrane detachment. It's much more difficult to treat, although the data on treatment and rebubbling to get this to go back, we have not published yet, but it's interesting and we're working on that. So here again, you can see post-surgery, a lot of persistent corneal edema. But look at this, there is a straight line and some wavy lines. So this is a mixed detachment. And here you can see there are clear 2 lines. So this is the pre-desmoid layer, and the thin arrow and the shorter arrow is the wavy desmoid membrane. So next time you look at desmoid membrane detachment, you will be able to see that most of them will fit into type 1, type 2, or mixed, just like with the bubbles. But the story doesn't end there. You can have these kinds of detachments with a tear or without a tear. This we all know. So here is a nicely scrolled type 2 desmoid membrane detachment, which is regmatogenous or torn desmoids. Here what you see is a mixed type, but the desmoid membrane is torn, but the pre-desmoid layer is not torn. And what you see in acute hydrops, you got both of them are detached, but both of them are also torn. So the tear has involved, so it's a regmatogenous mixed detachment, and that is the cause of acute hydrops. So, but we have never seen where only the pre-desmoid layer has torn, but not the desmoid membrane. That we have never seen, but both together or the desmoids alone, you will see quite often. And interesting that these are the features that are very consistent along all patients. Then the next very interesting part of the surgical anatomy, and this is going to be in the next decade a main focus of research, is that the pre-desmoid layer, it actually terminates into the trabecular meshwork. So the trabecular meshwork is a continuation of the pre-desmoid layer. So you see here, that's where the desmoid membrane ends, but the Duer's layer, the pre-desmoid, carries on, and then it opens up like a fan to form the trabecular meshwork. This is on a very low power of a transmission EM, but look at the scanning EM. The desmoid membrane has been removed, and what you can see here is the pre-desmoid layer, the Duer's layer, which is actually becoming the collagen core of the trabecular meshwork over here. And this is illustrated in this very nice way to show it is the scarf. This scarf is the pre-desmoid layer, and it is made up of the same fibers that are coming out here to form the trabecular meshwork. These are not different from what is in here, only here they're very compact. And if you look at the red thing, that could be the desmoid membrane, but then the pre-desmoid layer will cover all the desmoid membrane here, just to show you what the concept is. Now, interestingly, deep sclerectomy is a procedure which we may all know about, where you go so deep that you de-roof the canal of Schlemm, which is over here, and you expose the descemet membrane and you allow the fluid to ooze out from the descemet membrane. This is a diagram, and here on cross section you can see you make one flap, then you take this huge block away, and that's from the descemet membrane. Aqueous is coming and is forming this bleb. And this is how it appears in actual surgical images I've taken from the web. But this colleague of mine, Harshvardhan Ghorpade and Morekar, his colleague, they have shown, they work from India, that in cases where you do deep sclerectomy and you leave the Duer's layer behind, the outcome is not as good as when you take the Duer's layer away as well and expose purely the descemet membrane. So not many people actually make this distinction during deep sclerectomy. But what they showed was that in all the samples where they took the block out and the Duer's layer was taken out with the block, by scanning EM, they were able to show that it was out. The results were better than where the block they took out did not show the Duer's layer, which means it was left behind. And they were able to also show a video audit by putting trypan blue dye and seeing the aqueous ooze and dilute the dye in another poster that they have presented. So I think a very important area of glaucoma research is opening up in relation to the layer. And we are also doing some very interesting studies on this in Nottingham. So if you cut the layer, then it's possible that it causes some problems in the trabecular meshwork in terms of collapse or whatever, and it may not allow the pressure or the aqueous to drain normally. So this is based on the clinical observation that the incidence of glaucoma after DALC is much less than after PK. And in PK, this layer is cut. In DALC, the predescemet layer is not cut. And the difference in pressure cannot be explained by the use of steroids alone. So clearly there is some anatomical normality we are leaving behind when we do a DALC and preserve the predescemet layer, which is not there when we do a PK and we actually cut through the Duer's layer. So that's an area in glaucoma where further research you will see coming in the next 10 years. So just to go back to that little example, coming from the sclera where in the direction of the arrow, the flap will come like this, and that much part of the predescemet layer within that white bracket has to be excised, leaving the trabeculum so that the red underneath the descemet membrane can be exposed. So in deep sclerectomy, if you leave that there, then it's not going to work as well. Now, remember I told you that the predescemet layer is impervious to air. Then how does air get through it to form a type 1 bubble? So the answer lies in the fact that the type 1 bubble always, the type 2 bubble. The type 2 bubble is the one where air goes between the predescemet layer and the descemet membrane. So the type 2 bubble always starts at the periphery. So what we did is we marked the starting point, then we peeled off the descemet membrane and we looked underneath. And what you find is that there are about 15 to 17 clusters of minute holes that are present in the predescemet layer just before it starts to become the trabecular meshwork. And the average size of the hole is about 20 microns. So you can see if this is a very high power scanning EM of the predescemet layer becoming the trabecular meshwork, and there are some holes over there. So imagine that the descemet membrane is attached here. Now, if you inject air and air escapes through these holes, it will go anterior to the descemet membrane to form a type 2. Sorry, beg your pardon, let me reverse that. The air injected coming out from here will go into the anterior chamber, and it will form bubbles in the anterior chamber, as we see quite often when we do DALC. But if the descemet membrane is attached here, then the air will go anterior to descemet membrane to form your type 2 bubble. So you can see how this novel feature and understanding of the microanatomy of the cornea in terms of these holes and the clusters present at the periphery can explain why you get a type 2 bubble and why you get a type 1 bubble and why a type 2 always starts at the periphery. And on the basis of that, the clinical application was this. So when you see these holes and a lot of air is escaping, you don't want a lot of escape of air when you are doing or you're creating a type 1 bubble, which is the pre-Descemet's layer and the Descemet's membrane, to do a PDEC operation, which is the pre-Descemet's endothelial keratoplasty. And in PDEC operation, you also don't want a type 2 bubble to form, because you want the pre-Descemet's layer, not just the Descemet's membrane, because then it forces you to do a DMEC, which is not bad, but if you're doing a PDEC, you don't want that to happen. So what we said is if you clamp these holes completely with a clamp, then you should stop a type 2 bubble from forming and you should stop escape of air. So that's what we did. We actually invented a clamp to do exactly that. And you can see over here, there's the clamp. And again, this video shows that if it works, that we can actually, again, the video is not going to work here, inject the air through that side port, and it'll create a type 1 bubble all the time without a single bubble of air escaping. So we have applied the surgical anatomy knowledge to a clinical application by creating this clamp, which then helps us achieve our purpose. So here is the PDEC operation. You can see the bubble has been created. We're taking the PDEC tissue, loading it into a cartridge, and then you inject it into the eye. It's just like a DMEC operation. It also curls in the same manner, but it is a smaller piece of tissue, and because it's much easier to unfold in the eye, handling required is much less, and this is the outcome of a PDEC following 2 months post-operative. And there are lots of papers on this also published. The other interesting thing which we published right in the beginning, in our first paper, that acute eye drops is not due to rupture in the Descemet's membrane. Everybody has taught us acute eye drops is due to rupture in the Descemet's membrane. Actually, acute eye drops is due to rupture in the Descemet's membrane and the pre-Descemet's layer. This is what we said when we published our first paper, and then more literature has appeared to prove that. So here you can see it's a patient with acute eye drops in a graft, which was done for it's an eccentric graft, but you can see that you get a type 1 Descemet's membrane detachment. So the Descemet's membrane is there, and you can do further scans. You will see the tear, but the clinical application of this came from Morin's group. What they showed is you can actually stitch together by giving mattress sutures to the pre-Descemet's layer, leaving the torn Descemet's membrane aside. You don't have to stitch the Descemet's membrane. If you just stitch the pre-Descemet's layer together, you can reverse acute eye drops very quickly. So that paper was published in the BJO. But recently, Meles, who initially said that, it doesn't believe the layer exists, actually proved that it does exist, and he published this paper. So he was doing Bowman's membrane transplant, and in keratoconus, because the eyes are so thin, sometimes they penetrate through the pre-Descemet's layer coming from anterior posterior. And whenever they penetrated the pre-Descemet's layer in keratoconus patients where they were trying a Bowman's membrane transplant, they got acute eye drops. But when they did not penetrate that layer, even if the Descemet's membrane was detached, they did not get acute eye drops. So they actually concluded that tear in the pre-Descemet's layer is essential to have acute eye drops, which is exactly what we had said many years ago, about 7 years ago. Then on that basis, we wrote a letter to them saying, this is clinical evidence of what is difficult to do in vitro as an experiment, because you can't get eye bank keratoconus eyes to do that. But what we found is that if you try and incise the Descemet's membrane and the pre-Descemet's layer in an eye bank donor eye, put it in an anterior chamber and raise the BSS pressure up to even 60 millimeters mercury, you don't get acute eye drops. So it's not just the tear in Descemet's and pre-Descemet's layer, but you also need the abnormal collagen of the keratoconus eyes. So when all three come together, you get acute eye drops. And in our reply to our letter, they said that we thoroughly agree with the primary point of the letter, that acute eye drops was a result of tear in both Descemet's membrane and pre-Descemet's layer in the background of the abnormal collagen of the corneal stroma in keratoconus. So we're very grateful to them for having proven through their surgery that this tear in the predestined layer is important. So you can see how now we are beginning to understand corneal pathology better based on observations on the Duers layer or the predestined layer. And then that's not where the story ends. So if you look at desmatoceles, if you do OCTs on all your desmatoceles and look very carefully, you will find that at least 50% of the times, if not more, the predestined layer is still there. In fact, Sadir Hanush calls it a duocele. He says it's only when the desmus is exposed that you can call it a desmatocele. And the predestined layer, as long as it is preserved in the desmatocele, the rupture rate is low. But once the lytic process dissolves even the predestined layer, then the desmus membrane ruptures easily or perforates and you get leaks. So that's another story that is building up, and we will write about it, but already a case report has appeared from Mr. Amar Agarwal from Chennai in India. And just to finish off with one or two other points. So if you take the predestined layer and the desmus membrane together, so this is PDEC tissue, what you will see in a type 1 bubble. It scrolls very little compared to if you separate. Now, these are the same tissue from which predestined layer and desmus membrane has been separated. Same tissue here, they separate the predestined layer and desmus membrane. And you can see how much more the desmus membrane scrolls than the predestined layer on its own, which scrolls very little. So most of the scrolling characteristics of predestined tissue is by the desmus membrane. And this is due to the elastin, as everybody believes elastin will cause it to scroll. But when you look at the actual quantification of the amount of elastin, you will see that the maximum amount of elastin is actually in the predestined layer, even more than in the desmus membrane, much more. And almost the same as in the trabecular meshwork, which is because they're more or less the same tissue going through. And the other parts of the cornea and sclera have very little elastin. So it's not just the presence of elastin, because if it was only elastin, then this should scroll more than the desmus membrane. But we know that this scrolls more. So what else is there that we don't know? And this is the answer. So this is the predestined layer. It has a lot of elastin all through the layer. But this is the desmus membrane. And you can see all the elastin is concentrated as a band in the anterior part of the desmus membrane. And this is PDEC tissue, as you can see that band again, and this is the whole cornea. The whole cornea, you can see that's the desmus membrane with the band. The interfacial matrix is darker, no color, and then there's a nice band of elastin in the predestined layer and then the corneal stroma. So not only was this the first evidence that structurally the predestined layer is different from the rest of the cornea, but that the elastin distribution is very different, as you see over here, from what it is in the desmus membrane. So how does this help us clinically? Well, here's the story. So if you take a DMEC tissue and let it scroll, and then you put elastase enzyme on it, slowly the whole tissue will unroll in front of your eyes. We're not touching it even. The elastase enzyme will degrade the elastin, as you see in panel D. You can see over here, all the elastin is degraded. This is the normal elastin, how nice and dense and clean. Here it's all gone, and that tissue is completely opened and flat. So we are now looking at, it's possible to mildly treat with elastase for a short period of time, right temperature, right concentration, exposure, etc., so that it becomes easier to open in the eye without actually permanently destroying the elastin. So that's a potential application, but we're far from it yet. So the elastin band is what is causing this to happen. And some people say in the literature, it's the endothelial cells, the swollen endothelial cells that cause it to roll that way. But if you take desmus membrane and you take away, these are the endothelial cells, the dots, you take away all the endothelial cells by dispase, you will find it scrolls just the same. So the endothelial cells actually don't play any role in the scrolling, it's the elastase enzyme. So my final 1 or 2 slides now. So this is just a diagram to show, so God did not put this elastin band in the anterior surface of the cornea for us to do DMEC. It is there for a physiological purpose. And the physiological purpose is that you can imagine the desmus membrane has to apply to the back of a concave surface without any structural attachments. This plane of cleavage is very, very light, very easy to separate, but it has to be firmly attached to that so that it can do the pump action. So I'm not sure if you can see my hands, but if this is the back of the desmus membrane of the cornea, and this is the desmus membrane. And if the desmus membrane is now going to want to scroll like this, because it always scrolls with the endothelial cells outside, that elastin will keep it constantly pressed against the back of the cornea. And that's why it is there, and that's the role it is playing. So again, it explains the physiology, and it explains why DMEC tissue and PDEC tissue scroll the way they do with the endothelial cells always on the outside. So I finish with this summary. The Dues layer has improved our understanding of corneal surgical anatomy, of the DALC operation, other lamellar corneal surgeries, and of corneal diseases. It has made DALC a much safer operation. If you know which types of bubbles you've got, you can stop them from rupturing on you. It has led to the innovation of 3 new surgical procedures. One is the DALC triple, which is doing cataract surgery under the predesmus layer during DALC, the PDEC, which is predesmus endothelial keratoplasty, and suture management of acute eye drops. It is involved in the pathogenesis of posterior corneal disease. We saw desmetocele, clearly desmus membrane detachment, and once again, I acknowledge the help given by all the colleagues I mentioned, and particularly Rajesh Sinha, who contributed more than half the cases from All India Institute of Medical Sciences to prove that this desmus membrane detachment is not what we think it is. Acute eye drops, we've seen our understanding has changed. Macular corneal dystrophy, I didn't mention about it, but it's another area where this layer comes into its own, and in the spread of infection. And finally, which is going to be really a very big area, is role in glaucoma. Again, Dr. Ghorpade kindly lent me his data on deep sclerectomy and some of our own work on how the perfusion to the trabecular meshwork can change depending how pathology affects the predesmus layer. And this is being investigated. And I didn't even mention the role this layer plays in the biomechanics of the posterior cornea. It's another area of study. We know that keratoconus starts as posterior keratoconus. And already a study has shown, this is from Keith Meek's group, that the elastin degradation in the predesmus layer happens earlier on in keratoconus. So you can see that posterior keratoconus probably starts in the predesmus layer and as pathology of the predesmus layer. So there's a lot more to come. And despite all the glitches and all the hacking and everything, I'm really grateful for you, all of you, to give me this opportunity to talk to you. Thank you.
Okay, Professor Dua, it was a really amazing presentation. I think that it's very difficult to ask you a question because the presentation was so high level, it was perfect. But we're going to ask you some questions with Guillermo. Can you give us some tips to improve our outcomes in having more type 1 bubbles for our corneal surgeries? One or two tips.
Yes, so it's very difficult to that question. Everybody asks the question, but I'll tell you 1 or 2 tips that will work. So if you go as deep as possible in the central stroma, if you are in the peripheral stroma, there's more likelihood you will get a type 2 bubble. But if you go deep in the central stroma, then the depth is important. In eye bank eyes, it doesn't seem to be important, but in human eyes, it is. The other thing that I might suggest is that if you can attain high pressure, so remember, whether you're using a 27 or 28 gauge needle, the pressure at the tip of the needle depends on the force that you are exerting at the back. So when you're pushing a plunger of a 10 ml syringe or a 5 ml syringe or a 1 ml syringe, the force per unit area, which is the pressure of your thumb on the plunger, is more or less the same, but the pressure at the tip will be the most for a 2 ml syringe compared to that for a 10 ml syringe. So, but a 2 ml syringe has not enough air to keep that sustained pressure for a long time, because air will go out through the whole cornea. Some air will escape into the anterior chamber through the periphery holes, as I showed. So if you have a longer syringe with a narrower lumen, you can exert high pressure. So being deep and high pressure will allow you to get the type 1 more often than a type 2.
Perfect, thank you. And how did you manage the type 2 and type 3 bubbles when you have type 2 and type 3? How did you manage that situation?
So type 2 bubble, you can get a perfect DALC and very good outcome with a type 2 bubble. Only you should be aware that this can burst anytime. So once you're aware, so the first thing is, if it's a type 2 bubble and it's complete, then you will not do a paracentesis until you have let the air out. So you will take away the top half to 2/3 of the stroma, then you will puncture. But remember, when you puncture, there's so much pressure in the eye. And we always say before you puncture, do paracentesis, because that releases the pressure. But here you're not done a paracentesis. So you have to put a lot of Helon, even Helon GV, on the surface of the cornea after taking the top layer off, and then puncture through the GV. So even if by a fraction of a second, you slow down the escape of the air from between the descemet membrane and the posterior stroma, then it will not come and hit your needle that fast. So you won't puncture it. So once you've done that, then you can do your paracentesis and complete the DALC. But keep in mind that during the operation, once you've released the air and the descemet membrane has become convex, you still have to keep doing paracentesis repeatedly, not just one time, but I would say every 2 to 3 minutes during the operation, especially when you're cutting the 4 quadrants out. Keep releasing aqueous, because if the descemet membrane is flaccid, it will not burst on you. And then you can complete it quite nicely. For the mixed bubble, all you have to do is, once you've exposed the pre-descemet layer, again, you can put a drop of Helon with a sharp needle, just puncture the pre-descemet layer, all the air comes out, and that sector of the type 2 bubble, that component collapses. If it's very small, you don't bother, but if it's large, then you release the air by tiny puncture of the pre-descemet layer over the second bubble, which is the bubble between the descemet membrane and the pre-descemet layer.
Okay, thank you. How did you manage your perforations, your micro perforations, when you are performing DALC?
So if you have a perforation, even a macro perforation in a type 1 bubble, just finish the DALC as normal, put air in the anterior chamber. When you put air in the anterior chamber, no matter what operation you're doing, you must check the pressure within 1 hour or within 2 hours maximum, but 1 to 2 hours, and then release the air if the pressure is high, otherwise you'll get Oritz-Zavalier syndrome. That has happened with many people, and it is not nice when it happens. So bear in mind, air in the anterior chamber can cause high pressure. So that is what one would do. You don't have to convert to a PK with a type 1 bubble. With a type 2 bubble, which is descemet membrane, when you get a tear, it will extend. And if the descemet membrane is convex and stretched and you get a tiny puncture, it'll burst. So then you have to convert. But if it is a tiny tear, remember the descemet membrane will roll. And if it rolls, it's difficult to unroll. So those are more difficult to manage, these punctures in a type 2 bubble. Sometimes you get away with it, sometimes you don't. But with a type 1 bubble, almost 95% of the times or more, you can get away with it.
Okay. Guillermo, do you want to ask some questions?
Yeah, this is a very interesting lecture. I learned a lot personally. There is a very good question here from Nestor.
he's asking in inflammatory processes when you, for example, after infectious keratitis, once they're healed and you do a DALC, do you think there's a strong adherence between the stroma and Dua's layer, and this is why we get a lot of type 2 bubbles?
Yes, so the deeper the infection, in our experience and what we published in our first paper, and we got a lot of stick for that, is to say that this layer is practically devoid of cells or is acellular. And that is true in the normal situation. But like I showed you, even in type 1 desmoid membrane detachment, keratocytes can get into it. And in inflammation, once this layer gets inflamed and is part of the inflammatory process, then scarring will happen and that cleavage plane will be obliterated in places. And not only that, the layer itself, some of the elastin will be replaced by scar tissue, collagen. And then you will find that it's much more difficult to blow it out as a bubble. And that is why then the air is pushed out to the periphery, and then you get a type 1, a type 2 bubble. Also, you find in advanced keratoconus, what happens is that sometimes this layer is already stretched so much. You can see how far the cone has come forward. The layer is already stretched and the fibers are loose. And you will find that the air can push the fibers further apart and go in, and you get a mixed bubble. So in advanced keratoconus also that can happen. But usually when you get a mixed bubble, the type 2 component is coming from the periphery. Yeah, and that will happen. The person who asked the question is right in that when you get scarring, then you get more type 2 than type 1.
Okay.
But you know, I could just add one small point. Even if you get a very small, you know, when we talk about DALC, and we have a big wet lab on DALC, we show this. Even if you get a very small type 1 bubble, very small type 1 bubble, you can open it and put a spatula inside and separate the whole cleavage plane. So if there's a history of scarring in the background or some infection, and you get a type 1 bubble which is small, then you can go in and you can break those adhesions and then create it exactly like a type 1 would have happened with air. So just bear that in mind. Sometimes you get lucky.
Okay. What about the you show us the different types of the Descemet's detachment. Is there any clinical implications in your algorithm of management of these detachments when you have each one of the detachments?
Yes, I will tell you that. So it is not part of the paper, which, like I said, was just accepted, but it is something that will come. And if you have a type 1 desmoid membrane detachment, the outcome will be not as simple or not as good as with a type 2 detachment without a tear. And the difference will be the sooner you detect it and treat it. So if you get a type 1 detachment and you're treating it within a week or 2 of its happening, you can get a good outcome. But once the contracture starts and it starts to shrink, the line becomes even like the string of a bow, you know, it's very tight. Then if you put the air, it will go back, and as soon as you release the air, it'll come back. So you might have to do a desmetectomy just to release the attachment at some place so that it can go back and stay back. So you will find the duration of the type 1 detachment will determine how easily it will reposit just by air injection. So all these things we will now learn more and more of, because the longer it stays and cells infiltrate, the more scarred it gets.
Yeah. I have another question here from the chat. Dr. Francisco Sanchez is asking, which is the limit of trying DALC in keratoconus? Sometimes after eye drops, the air breaks the Descemet membrane.
Yes, so when you got after acute eye drops, remember, both the layers are torn. So that is the concept which not many people knew before. They think only Descemet is broken, so the type 1 bubble can still happen. But no, after acute eye drops, both are torn. They are replaced by a very thin Descemet membrane, secreted by the endothelial cells which are closing the gap. So when you put air pressure, you will find you will be very, very lucky if you get a bubble at all. Chances are it will go through. So you can try a mechanical dowel. In fact, I don't try a air injection dowel following acute high drops because I know it's going to go through. But mechanically, you can go and in the area of the original tear, which you can see on OCT, you take your dissection such that you leave a bit of stroma behind. So that's the only way you can do a DALC. But if that stroma you're leaving behind, which is essentially scar tissue, is in the visual axis, the outcome is not so good. So PK might be better.
Dr. Dua, I have a question that may not be 100% related to the topic, but I want to pick your brain. When we do keratoprosthesis or multiple penetrating keratoplasties in this retrocorneal membranes form, the retroprosthetic membranes, and that we also see in patients with multiple failed DSECs and disorganized or sick anterior segments, do you think there is a role for this for Dua's membrane in the pathogenesis of these membranes?
So for retrocorneal membranes, I think it's definitely, in some instances, it might be that it's this membrane, this layer itself that has separated and got scarred and infiltrated with cells. And we're calling it a retrocorneal membrane, but it may be that it was just this layer. But retroprosthetic membranes and cyclitic membranes, they are different because they come from ciliary body exudation. I said behind the K-Pro, for example, they are not this layer. But a retrocorneal membrane, it certainly might be that in some of them, this is the only layer or membrane, or it is a component of the membrane that has formed. We will know now that we know, we'll do histology. Everybody will do histology to look at retrocorneal membranes in more detail.
Yeah, because some of the retroprosthetic membranes, there's some data that it may be stroma that's migrating. And this is why the use of larger diameter plates was recommended. But you know, I personally didn't see the benefit clinically. So I don't know if the Dua membrane is functioning as a scaffold on retroprosthetic membranes.
But we've got a hole. See, we've got a hole in the membrane. And that is, so the collagen on its own will not migrate. The keratocytes will migrate, and they will lay down abnormal collagen. So if you have a plastic piece protruding through the back of the cornea, which is what a prosthesis does, then the keratocytes, and they become, you know, as you know, myofibroblasts and fibroblasts, and there are a lot of other inflammatory cells as well, and they can then migrate and create a scar tissue, which we then call the retroprosthetic membrane. So it will be, to insert it in place, we have made a hole in the whole corneal stroma, including the predestined layer.
Okay, thank you.
Okay, I think that we are going to close the webinar. You want to do another question, Guillermo?
No, I mean, I think well, there's one last question from.
Okay, maybe I will ask a personal question to Professor Dua. When you realized that you have discovered this new layer of the cornea, who was the first person who knows this? Who was your confidence about this?
Well, we were working as a team. So the first individual whose name doesn't appear in any of this is a lady called Anupama Pherwani. She was my fellow at the time, and I actually gave it to her as a thesis project. I said, look, there is definitely this membrane. We have this. It was then an idea. There were the clinical clues were there. I said, let's work on it. And this was way back in 2007. As you know, some of the slides I showed were electron microscopes from micrograms from 2007. But for some reason or other, she couldn't do it, although the concept was first discussed with her. But then when we actually discovered the layer, and I showed you that slide, you know, when we peeled off the Descemet's membrane from the bubble and showed that the bubble is still intact, you know, that was what I call the eureka moment. And I think it was Dr. Dalia Said, who was then there when she first said, "Oh, this is it." We first proved that, and I call that my eureka moment. Whether the definitive proof was there at that time. But as usual, it went through all the stages. First there is disbelief, then it may be so, then people say yes, and then they start producing even more and better evidence of its existence than I did. So every new thing goes through these cycles.
Okay. It was easy to publish the paper?
Actually, the paper, we tried to publish it in a New England Journal of Medicine. They said no. We tried to publish it as a letter to Nature Medicine. They said no. Then we sent to ophthalmology, and I sent as a pre-publication request to them saying, this is the idea, and what do you think? So I don't remember his exact words, but the editor was quite supportive. He said that this is a very bold, courageous idea. Well, let's see what the reviewers think. You can send it. So I sent it to them, and then it was published with all the videos. But the interesting thing is that ever since that paper in 2013, except for one, any paper I have sent to ophthalmology related to the layer has been rejected outright. Some not even with review, because one of the editors wrote a very scathing editorial in that issue when it was published. He didn't mention my paper at all, but it was pretty clear that it was directed at the paper. And none of we have published maybe at least a dozen more papers since then, and at least 4 of them were sent to ophthalmology first, and they were rejected. Then I stopped sending to them, and AJO has been very good. And even the 2 papers that were rejected by ophthalmology were published by AJO. I told them they've been rejected by ophthalmology, and these are the reasons they have given, and I have addressed those reasons, and both were published in the AJO, as is now the DM Descemet membrane detachment paper. So I didn't send it to ophthalmology, I sent it straight to the AJO. And even the next paper on desmetosceles, for example, again in collaboration with colleagues in India, they get a lot of pathology, a lot of papers, and very good with their clinical examinations. So we will not send to ophthalmology, stop sending, because at the end of the day, it doesn't matter where it's published, it's what it is saying and how many people accept it and cite it, which is more important.
Okay. Guillermo, the last one, do you want to do another question?
No, I mean, I wanted to ask, you asked the question in a very more formal way. I wanted to ask how many times the paper was rejected, because sometimes I feel rejection, as a young physician trying to publish, it discourages, but some of the rejections, if you keep trying, those are the ones that come and change the field. You have publications that, there's many Nobel Prizes that have been denied for publication, and then later on they come and change the field. So this is, congratulations, Dr. Dua. This is very,
thank you.
Very good.
When you publish something new, there are, I tell this, there are 4 types of reactions. One is, it can't be true. The other is, it's been described before. And third is, I've described it before. And the 4th one is, let's check it out and see. And fortunately, the 4th one is the majority. But often one of the first 3 can be the ones that influence the decision of publication or rejection. But most of the people say, well, let's keep an open mind, let's check it out and see. And then when they check it out and see, and it bears the scrutiny of your peers, then it becomes the landmark that it deserves. So every paper goes through this, and one has to, take it in the stride. It's disappointing. I agree with you entirely. Even for an established researcher, never mind a young researcher, it is disappointing.
Okay, we're going to close the webinar. Thank you very much, Professor Dua. Your presentation was really amazing. We have a good time with you and Guillermo. I hope we can meet in any meeting and continue talking about cornea and corneal surgery. That is what we like. Have a good day and stay safe of hackers.
Thank you very much. Thank you.
Thank you.
Bye-bye. Thank you.
Bye. Bye-bye.
Bye, Ticiano. Saludos.